Okay, so we all know smoking is bad for you, right? We've heard it a million times. But what if I told you it leaves a literal physical signature on your DNA? Not by changing your actual genes, but by messing with the instructions for how your body reads them. Let's dig in and see how that actually works. And that's the big question we're tackling today. We're going to unpack a really fascinating study that discovered specific, measurable changes happening on our chromosomes. And they're all directly linked to smoking cigarettes. Right, so here's the key thing to get your head around. We're not talking about mutations, rewriting the actual code of your DNA. The changes are happening in a different layer, a kind of control system that sits right on top of your genes. This entire control system has a name, epigenetics. So imagine your DNA is a giant instruction book. Smoking isn't rewriting the words in that book. Instead, it's going through and slapping on these little sticky notes that tell your body which parts of the book to read, which parts to ignore, and which parts to read over and over again. Now, the specific type of Post-it note we're looking at is a process called DNA methylation. The best way to think about it is like a dimmer switch for a gene. And what this study found is that smoking can literally get its hands on that switch, turning a gene's activity way up or dialing it way down. So you'd think scientists would have this all figured out. But for the longest time, they knew smoking was affecting our epigenetics, but they were missing a massive piece of the puzzle. They were basically ignoring an entire chromosome. And that missing piece? It was the X chromosome. For years, researchers just left it out of these big studies because, frankly, its unique biology made it a complete nightmare to analyze. It was a genomic black box. And this illustration shows you exactly why it was so tricky. See, males have one X chromosome, so you get a nice, clean data signal. But females have two, and in each cell, one of them is randomly shut off. This creates that messy, two-humped signal you see on the right. One peak for the active chromosome, one for the inactive one. For scientists, trying to untangle that was a statistical headache. So how on earth did the researchers in this study finally crack the code? Well, they came up with a really clever new method, one specifically designed to handle all that complexity of the X chromosome. Their approach was actually pretty brilliant. First, they just analyzed males and females separately, which, boom, gets rid of that messy signal problem. Then they did a deep dive, scanning just the X chromosome for any smoking-related changes. And this is so important, they confirmed everything they found in 3 other independent groups of people. And you know what? After all that careful work, they found it. A clear, specific, and replicated signature. Smoking was consistently flipping the dimmer switches on 2 particular genes located on that overlooked X chromosome. All right, here's the first finding. Take a look at this top graph. As you move from left to right, from people who've never smoked, to past smokers, to current smokers, you see that value on the left just steadily climbing. This is called hypermethylation. Smoking is adding these little marks to the BCOR gene, and it's like it's turning that gene's dimmer switch way, way down. But here's where it gets really interesting. Smoking doesn't just turn things down. In a different gene, called TSC22D3, it does the complete opposite. It actively removes those marks. That's called hypomethylation, and it's like smoking is grabbing that dimmer switch and cranking the gene's activity all the way up. So let's just break that down one more time, nice and simple. For the BCOR gene, smoking adds marks, which turns its activity down. For the TSC22D3 gene, it takes marks away, which turns its activity up. 2 different genes, 2 totally opposite effects, all from the same cause. But wait, there was another fascinating twist. The data showed that the effect on that BCOR gene, the one getting turned down, was about twice as strong in males as it was in females. So why? Well, it goes back to that biology. Since males only have one X chromosome, it takes the full epigenetic hit. In females, with one X already silenced, the effect gets kind of diluted. And if you're thinking, okay, but maybe this was just a fluke in one group of people, this chart right here is the proof it wasn't. This is called a forest plot, and all you really need to know is that when all those little squares line up on the same side of that center line, it means the result is solid. They found the same thing again and again and again. It's real. Okay, so smoking flips a couple of genetic switches. Big deal, right? Why should we actually care about these tiny little marks? Well, the answer is all about what these 2 genes actually do in our bodies. Yeah, these are not minor players. BCOR is what's known as a master regulator. It basically helps tell a whole bunch of other genes what to do, and it's critical for how our stem cells develop.
And TSC22D3? It's involved in huge things like our immune system, how our bodies create fat, and even kidney function. Messing with the dials on genes like these, yeah, that's a big deal.
The study's authors really put it perfectly themselves. By finally figuring out how to study the X chromosome, they're saying we've opened a brand new door to understanding how the things we do, our environment, interact with our genes to affect our health. So, if you take just one thing away from all of this, let it be this. Our lifestyle choices don't just affect our health in some vague, abstract way. They can leave a specific, measurable, biological signature on our very own DNA. A signature that science is, right now, finally learning how to read.
And that just leaves us with one huge final question, doesn't it? If smoking leaves this distinct signature on our DNA, what else does? What kind of marks are being left by our diet, our stress levels, or the air we breathe? That is the next incredible chapter that science has to explore.